be talking about joint protein sequence structure co-design by equivariant diffusion. That's work been done with my collaborators and advisors, Kevin Yang and Lauren Crawford. So if you took a high school biology class, you might remember that proteins are the biological machine. But if you think about probably some of the more important and interesting technologies in our everyday lives, like PCR tests, antibody development, and enzymes to break down toxic substances, treat water, etc., they're all powered by protein engineering. So the question kind of becomes, if we know what protein functions we're interested in, then what exactly needs to be designed? And broadly speaking, we have two fundamental ways of looking at proteins. We have sequence and structure features that give rise to these more complex tertiary quaternary structures based on their interactions. So from these two features alone, we can glean a lot about evolutionary history, function association, physical attributes, features, etc., that are associated with particular proteins, domains, and families. So the classic design problem really boils down to generating a protein sequence or structure that performs some function, which is basically through conditioning on some of these proteomic features that I just mentioned. And you can typically, in one aspect of the problem, either generate a sequence or a structure. But in this work, I'll be showing a method to co-design proteins, which reduces a lot of the error involved in this back and forth inference process. And the idea is that we can jointly generate functional protein candidates. So we've seen a lot of success with diffusion models sort of in text and vision domains, and also increasingly more in the natural sciences and structural biology domains. And I think generative models and diffusion models in particular are really powerful to generalize over some distribution and features of interest that we can condition on. So in our case, we'll be looking at proteomic features. And the core idea of the diffusion model really is that you start with this perfectly described distribution over some variable. So in our case, we're looking at protein sequence and structure features. You iteratively add some noise, approach this fully noise distribution, and then over time learn to predict the mean and variance at each time step to denoise the distribution. And then from our fully denoised distribution, we can sample to obtain a design candidate of interest. And so really we're learning to reconstruct protein sequence and structure samples in an autoregressive way. So when we think about co-designing proteins, we're thinking about designing sequence and structure features together. So when we think about the information or the data that we're gathering, we're looking at the C alpha backbone coordinates that we construct this probability distribution over our variable x. That's our coordinate data. And then we have the corresponding set of amino acids for those particular structures. We put that through a pre-trained language model, and then we can obtain a latent sequence distribution. And so the idea is that we can also have a really nice combination of continuous and discrete features of proteins by doing this sort of joint approach. And so from these two sets of features, we can construct this graph. So we combine X, which comes from our structure data, we combine S, which comes from our sequence data, and then we add in this H, which is our positional embedding, and those three variables concatenated gives rise to each individual node in this graph. And then this particular graph is a representation of one protein that we can use a collection of these graph structures to diffuse over for a diffusion model. And so we take this structure, which is our input to our equivariant diffusion model, put it through, so we're iteratively noising and then denoising, and then we learn the final reconstructed distribution, and then we can sample from this distribution to obtain a corresponding set of sequence and structure for a particular protein. So when we think about results or evaluation, we kind of need to think about evaluating our structures in this forward direction, that is sequence to structure, and then also the backward direction of structure to sequence, because we're generating these candidates jointly, so we need to evaluate them jointly. So when we think about sequences, we can take our generated sequence, which is our seq star, put it through AlphaFold 2, which we get an AlphaFold prediction, which we treat as ground truth, compare that to the generated structure that came out, calculate this TM score, and then we get this self-consistency TM score graph for sequences in particular. So we see that a score of greater than 0.5 is what we consider to be designable structure, and we show that we can recapitulate a little bit of that distribution that the orange distribution is showing. And then for structures, we have a generated structure. We put that through protein MPNN, which is a structure to sequence model, obtain a sequence prediction that might fold to that structure, put that through AlphaFold 2, and then again we do the same thing where we compare our ground truth structure to our generated structure, and then here we can again show that we recapitulate some of this blue distribution of over a 0.5 value, which is a designable structure. But I think the more interesting question is, how can we co-design these proteins and compare them to natural proteins in terms of function. So we have membrane solubility and subcellular localization as two sort of proof of concept function validations here. And we again show that we recapitulate some of the same distribution that we observe in natural sequences. And the goal is to be able to generalize to more functions and more features as well. So thank you for listening, and you can click on the link in the description for more links to the paper and to the code as well.